Our next speaker is Brandon Brown from UPenn CHOP team, who will also be discussing in STXBP1 AAV gene therapy.
Does it matter which curve? Jim, thanks for the introduction and the invite to speak. As Jim mentioned, I'm a postdoc at CHOP in Bev Davidson's lab, and all the work you'll see today, or most of it, is part of the END group. A little bit of overview. I want to make sure we're all on the same page about AAVs, how we think about designing AAV-based gene therapies, and then we'll go into where we are at with our in vitro and in vivo testing with our developed program here. Before I get into any science, while I'm presenting, these folks all have their hands in this work pretty heavily, so thank you to them, and just realize if you don't see faces on individual slides, these people probably had a role at some point in those experiments. So if you're not familiar with AAVs, they're pretty simple viruses, relatively. There's really only two pieces to understand here. We have a vector, or the DNA that's encapsulated in the virus to express our RNA and ultimately protein of interest. And there's a protein shell called a capsid, which will influence which cells that viron is able to produce and get into, and then also how that viron is intracellularly trafficked once it is into those cells. There's some wonderful things about these. We can modify them in the lab to be more cell-specific for targeting. They are non-replicating. They express no viral proteins, and there are currently 8 FDA-approved, really 9, one is off-market, AAV-based gene therapies. A couple of downsides to note. High systemic doses can cause adverse events, and they do have a limited packaging capacity of 4.7 kb, not really an issue for this program, though. So when I think about designing AAV-based gene therapy, I think about this in 3 large puzzle pieces. First is, what is our therapeutic strategy? We've taken the same strategy as we saw in the last talk. We know that the biologic need here is to increase STXBP1 protein levels by providing healthy copies of the gene. Our second puzzle piece is a delivery vector, or can we efficiently target the correct cells that need that gene of interest? Preferably with low peripheral exposure in this case. And cargo design, what should we deliver? Is it feasible to package? And ultimately, can it drive STXBP1 expression? I won't get into it, but there are interplay between all of these pieces that can influence how efficient one piece is relative to the other. Since we really understand the biology, I'm going to start with the delivery vector, or how can we target the cells of interest? Our lab takes the approach of screening for AAVs with enhanced CNS potency through large peptide libraries. All this work has been done by previous and current lab members that you see on the left. We start with random capsid sequences of about 4 million or 40 million capsids. These are infused direct to the CNS, either to the CSF or directly to the target tissue. In a relevant biologic organism, in this case, non-human primate. After that round of injection, the target tissue is collected. We refine that AAV library, and we re-inject that refined library with the same route of administration, recollect the target tissue, and we now have an enriched AAV library. Again, to drive this home, that takes us from 40 million capsids initially to 1 million in the first round to tens that go on for further in vivo validation. So using this pipeline, I have a few images that I think are really striking of how robust these capsids are that have come out of this pipeline. So we're looking at primary cortical neurons from a mouse in a dish. The top there in red, you're seeing AAV1 and AAV-PHP-EB. And the next set of images that will pop up is the first time in Bev's lab that I jumped from the microscope, ran down the hallway, and went, somebody else come check this. I don't believe this. You check the microscope settings, and sure enough, these capsids can really drive expression in neurons. In the next slide here, you'll see some NHP images. from another capsid development pipeline in the lab, again, just to reiterate how robust these capsids can be. So what we're seeing there is primary motor cortex and premotor cortex in the top right of those images, following a deep brain injection at a very low dose, which you can see on the bottom right there. We're able to target the relevant circuitry for this disease. So take-homes from our capsid development is that these neurotrophic capsids are far more potent. We're 10 to 1,000 times lower doses to hit the target cells. This broadens our therapeutic range so that if we do need to deliver more virus to have efficacy, we can do that hopefully safely. This lowers the cost of goods, ensures that we can scale this to treat patients hopefully, and also lowers peripheral exposure, improving safety because we are going directly into the brain. Maybe. All right, on to our third puzzle piece of our cargo design. What do we deliver and is it feasible and can it drive STXBP1 expression? We started with 3 cargos, all that have differing 5' and 3' regulatory elements. These are packaged into our developed capsids, which we just looked at. And we started with a dose of 5E4 in dual SMAD-derived iPSC neurons from the stem cell core. This is a moderate dose that you'll find in the literature for these type of neurons. And you can see that cargo 2 really drives STXBP1 expression in these C663 cell line. Again, well above wild type levels at a dose that's commonly used in the literature. Testing in another R367 stop line, we can see that cargo 2, which is now on the right in that Western blot, again, drives STXBP1 expression well above wild type levels. So we've gone on to do dose response, at least in these cell lines, of how low of a dose can we actually give to get back to wild type levels. Here's a Western blot from that, where 14C is the corrected patient line, escalating doses and quantification of that. You can see that 5E3, so 2 log folds lower than is typically used in the literature, we're getting back towards wild type levels. The group has also developed NGN2 iPSC-derived neurons here with the C663 mutation. My hands don't touch these besides feeding them, so thank you to those in the lab that make neurons look like this every time I pull them out of the incubator. As a neuroscientist, there's no more joy than when your neurons look like this. So again, we've tested this optimized cargo and NGN2 model as well. Again, we see the same thing, that somewhere between 5E2 and 1E3, we're getting close to wild type levels, again, roughly a log fold lower than what is typically used in the literature. And the last cell model in a dish I'll show you are mouse cortical neurons, isolated from P1 STXBP1 haploinsufficient mice. Again, a wonderful dose response where 5E2 gets us back to wild type levels of STXBP1 protein. Again, which is pretty remarkable that we're able to see this across in vitro models that were at least a log fold lower than is typically used. So these vectors are able to drive upregulation, at least in a dish, at pretty low doses. Does this improve neuronal function? The way we've gotten at that is calcium imaging, which we've seen in a few talks today, where if this will play, the brighter the neuron gets, the more active the neuron is, maybe. There we go. And if we turn this into a graphical representation of this activity, we're looking at raster plots here of an imaged well at day 17. And on the y-axis, you'll see neuron ID, so each row across this is an individual neuron that was identified. And we're looking at a 60-second video. If you'll notice there are vertical lines. Indicate that the neurons are firing synchronously. We can see that in an untreated STXBP1 condition, there looks to be more activity and less synchronized activity. And with treatment, back to wild-type levels of protein, this seems to improve network coordination. So just like the Jeffersons, we're moving on up. If anybody laughed, you just aged yourself. The students never laugh at those jokes. So moving in vivo, we've taken one of our peptide-modified capsids in our optimized cargo, bilaterally injected into P1 mice for a total dose of 2E10. And all of the data you'll see was from tissue harvested at 4 weeks. This is just to show that we do see haploinsufficiency at the RNA level in the cortex. We can see that at that dose, we are well above RNA levels, or wild-type levels of RNA. And if we also look in the hippocampus, maybe, we see the same thing in the hippocampus. So what happens to protein? As expected here with those pretty large bump in RNA, we do see protein rescue back to wild-type levels in the cortex. And we see the same story here in the hippocampus. So is this protein functional, and can it stabilize its binding partner, syntaxin? The answer there is yes. We do see that bump in syntaxin, at least with this small n that we're looking at here, back to wild-type levels following treatment. Since we have a good grasp on these 3 pieces of the AAV gene therapy puzzle, our next step is to go into efficacy testing. The way we're going about this is we use a multi-electrode intracranial EEG setup. We custom make these setups in the lab. We implant bilaterally to the regions that you're seeing marked there, and we record for up to 72 hours. As you can imagine, recording at 20,000 hertz for 72 hours from 10 electrodes from a live animal is a lot of data to process. So we've developed a pipeline that is pretty user-friendly for the non-computational neuroscientists like myself to get robust data out of this and hopefully standardize some of these analyses in the field. And here's just an example of readouts from this. We're just looking at motor cortex here, but as we've seen in other talks today, pretty robust seizure phenotypes, at least relative to spike wave discharges, which we're seeing there. And with that, I just want to thank everybody in the lab again. Those in bold are ones that directly contributed this to work, past lab members as well. Obviously, somebody has to pay for the science, so we're very grateful for that as well. And I'll take any questions.
Thanks so much. This was, again, really great, and you're going to say that you cannot say anything more, but I'm really intrigued in the 5' and 3' UTRs, of course, that you used. Can you say whether they were endogenous to the STXBP1, or is there any comment that you can make on specificity of these?
Ooh. Go ahead. So the most robust one is CAG, very large, strong promoter. And the 3' regulatory element is WPRE with poly A attached to it.
Right, okay,
yeah.
so, yeah, great.
We took some other strategies that we thought were interesting and pretty innovative, especially in the 3' UTRN, which I'm happy to tell you about, but just didn't upregulate as well. Yeah.
Yeah. Fantastic results, thanks.
Yeah, thank you. Cool. Thanks, Jim.